So in chromosome number 6, we have a gene known as human, as it is present in all human species. Like it's differ from species to species. Leukocytic means it's related to WBCs, and antigens, HLA antigen. These are antigenic proteins. So HLA, human leukocytic antigen, we have genes on the chromosome 6 which are clustered together to decrease the crossover. This is again an MCQ, to decrease the crossover rate. So this is present on the short arm of like this is a short arm and this is a long arm, and we have here chromosome number 6, and the chromosome 6 has the HLA gene, and in this HLA gene we have 3 segments, like class 1, class 3 and class 2. And this HLA gene, it gives major histocompatibility complexes 1 and 2. So major means there is also a minor one. So major histo mean tissue, compatibility means it is compatible to the tissue's complexes. It is a complex. Complex means it involves different structures. So it has 2 types, 1 and 2, and we will discuss side by side. So we have HLA gene, HLA A, B, C, HLA D, and on the D we have M, O, P, Q, R, and also its sub categories. We have B subcategory category of B 27, B 57, etc, etc. So these are all the HLA genes subtypes present on the chromosome number 6, and which encode the major histocompatibility complex. So in this major histocompatibility complex, first there are 2 types of complexes, 1 and 2. So let's discuss MHC. First discuss the differentiating features or special features of 1 and 2, then I'll give you some summary regarding how they act. So MHC1 and MHC2. So MHC1 mean it's related to either A, either B, either C, one letter. 2 means it has 2 letters, HLA, HLA-D M, HLA-D O, HLA-D P, HLA-D Q, HLA-D R, and their further subtypes. Then we have MHC. So it's 8 into 1 is equal to 8. So MHC1 is related to CD8, and CD8 is a cytotoxic cell. So MHC is related to ABC, and it is also related to cytotoxic CD8 cells of the T cell. Then we have MHC2. We have 2 letter genes, and then we have 8 is equal to 4 into 2 MHC2, 4 for CD4. It means that it involves helper T cell of CD4 category. Then we have HLA in MHC1. It is related to all the cytosolic proteins, and the MHC2 is involved the phagosomal pathogens, Phagosomal pathogen or phagosomal pathogen first. What is this? So all the viruses, antigens, viral antigens, all the cytosolic proteins, all the some bacteria, so some intracellular pathogens which replicate in cytoplasm, into the cytosol, they present they use MHC1 and CD8 response, and in the intracellular bacteria category of cytosolic replicating organism, we have a common example of listeria. In contrast, we have the pathogens, the intracellular pathogens, especially bacteria. They intracellular pathogen, they all bacteria use CD4 response, but there are especially two intracellular replicating bacteria like tuberculosis, legionella. They replicate in phagolysosome. So that's why these two are the only intracellular bacteria which replicate in phagolysosome. So they are using CD4 or MHC2 complexes. So these two are very differentiating MCQ. Afterwards, we have some pathway regarding this the pathway, like the bacteria, the MHC2, they use endosomes, and the MHC1, they use rough endoplasmic reticulum. The MHC1, they use a tap transport associated transporter associated cytosolic protein transporter, sorry, antigen processing. So this is a transporter associated with antigen processing. This is a protein which is used in MHC1, but in MHC2 we have this just the organelle, and it does not use any this kind of transporter. So no tap transporter associated with the antigen presenting. And then we have rough endoplasmic reticulum tap, and then it use intracellular it use endogenous antigens, endogenous antigens like intracellular bacteria, listeria, intracellular viruses, intracellular cytoplasmic protein. And the MHC, it use exogenous antigen like toxic shock syndrome, toxin, bacterial proteins, etc. The MHC2 is present, MHC2 and MHC1, these all are present in APCs. APCs like dendritic cells, B cells, macrophages, then we have Langerhans cells. But the MHC1 is also present in all the nucleated cells, not RBC, because RBC is a enucleated or non-nucleated cell, and all nucleated cell including platelets, and this is not this is only present on APCs. Then we have its structure. So regarding its structure, MHC has a long alpha 1 2 3 structure and a short beta 2 structures, and this beta 2 microglobin, it's present like this beta. But in the MHC MHC2 we have these are all the long and short mean they are variable or variant chains. We can say that these are variant chain, but in the MHC we have invariant or same. Invariant mean no variation, same chain, same in length, like alpha 1, alpha 2, beta 1 and beta 2 equal, like this. And next we have discussed almost all the differentiating features of these two, MHC1 and MHC2. So now we discuss the pathway. So regarding the pathways, we have MHC, like how an MHC1 and MHC2 work. In MHC1, this is a cell, it get infected, and the infected virus or infected cytosolic protein or infected self antigen even, or even a new antigen or a cancer cell, it infected this cell. So this antigen goes into the cell. Now in the cell it undergoes intracellular cytosolic replication, and this intracellular protein, virus, antigen, whatsoever it is, this antigen is actually it is used by ubiquitin ligase enzyme of the ubiquitin protease pathway, which degrade these proteins or these peptides, these polypeptides into small fragments. So these fragments goes to rough endoplasmic reticulum, and in rough endoplasmic reticulum we have a transporter associated antigen processing. This transporter take up tap in rough endoplasmic. This transporter taken up by the tap, it goes into the rough endoplasmic reticulum, and this tap is in close contact with MHC1, and this antigen activated this MHC1, and it goes then it goes to the Golgi apparatus, then to the granule, like secretory granule, and to the cell membrane and exocytose, and then expressed on the cell, and then the T cell, with respect MHC1, the respective T cell get in contact, bind to it and activate it and give the adaptive immunity response. But so this is all the mechanism of MHC1, but in MHC2 we have an exogenous antigen, and this antigen come into contact with this antigen is actually endocytosis. This antigen goes into the cell endocytose, This maybe immune evade lysosome, and this evasion leads to its intracellular growth. So it's evading lysosome and replicating it. When it replicate in the lysosome, then the lysosome or the endosome activated, and the endosome is actually the endocytosed part. And when the lysosome, like endocytosis, give rise to endosome, this having attached or combined with lysosome, acidified acidified and then attached. This lysosome having the MHC1 cells, MHC1 complexes inside them. This acidified lysosome has MHC1, which attached to this exogenous antigen. Then it goes to the Golgi apparatus, secretory granules, and then expressed, and then exocytosed, and then expressed on cell, and then it expresses MHC2, which recognized by CD4 helper cell, and it releases cytokines and give our adaptive immunity. So this is all about the MHC1, MHC2, HLA, etc, etc, etc. We differentiate these two features and this. So now there is an association regarding some diseases. So in some diseases we have the HLA associated. In 2021 version of first aid, they just reduced the number of HLA diseases and number of HLA gene. So it's very much difficult, like they skip A3, they skip B8, they skip DR2, DR5. So now we have HLA B27. B27 is very much important, with the seronegative arthropathies, including the pair mean reactive arthritis, and then we have ankylosing spondylitis, then we have psoriatic arthritis and IBD associated. So pair mean P for psoriatic arthropathy, IBD associated arthropathy. Then we have A for ankylosing spondylitis, and then we have reactive arthritis. And there they just introduce a new B57, and this is associated with the disease of Rebecca-Ware hypersensitive, and this topic is related to HIV medication and in the microbiology section. Then we have B27, so these are the B. Then we have the DQ2 and DQ8. So DQ2 and 8 mean eating. So eating is related to C and D. So this is for DQ, C for celiac disease. Celiac is eating related problem. So DQ2 and 8. And then we have DR3 and DR4. So in 4, one room has 4 walls. So room in rheumatoid arthritis, and DR4. So one room has 4 walls, so it is rheumatoid arthritis. And then DR3, so diabetes mellitus 1 plus 3 is equal to 4. So diabetes mellitus 1 is associated with DR4 and DR3. So diabetes mellitus type 1. And then we have the 1 2 3 2 3 and SLE is the old mnemonic. So now 2 is out of the picture, so SLE is DR3. And also there is an Addison disease, which is Addison's disease, which is associated with both of the HLA D3 and D4. So this is all about HLA associated diseases and HLA associated diseases. We have discussed MHC, we have discussed HLA, we have discussed the differentiating features between MHC1 and MHC2 and innate and adaptive immunity. Next I will tell something about bone marrow transplantation and an HLA mismatch, which are very much tested and it's very much confusing.